They say autophagy can be switched on by simply skipping meals. And now a newer claim moves through health culture like a shortcut, that certain foods can do the same thing without fasting at all. But inside the body, autophagy is not a trend. It is not a hack. It is not a button. It is a cellular recycling system, an ancient cleanup process in which worn proteins, damaged mitochondria, and biological debris are broken down and reused. It helps cells survive stress, maintain order, and slow the buildup of internal clutter over time. Fasting is one of the clearest known triggers because nutrient scarcity changes insulin, mTOR, and AMPK signaling. Food, however, enters the story differently. Some foods contain compounds that appear to influence autophagy-related pathways, but they do not replicate fasting in a simple or identical way. Source is source. So what do these foods actually do inside the body? And where does science draw the line between promise and myth? The failing cleanup. This chapter explores the initial physical signs of reduced cellular housekeeping. Autophagy is often discussed only in the language of longevity, but before it becomes a theory about aging, it is a practical act of maintenance. Inside each cell, proteins bend, fold, wear down, and sometimes misfold. Mitochondria, the small engines that turn oxygen and nutrients into usable energy, accumulate damage. Membranes oxidize. Waste builds quietly. Autophagy is the system that gathers part of that damaged material, encloses it, and delivers it to lysosomes, where it is broken apart and recycled. When that cleanup slows, the body does not announce it with one dramatic alarm, and the signs are indirect. Metabolic systems become less efficient. Cells carry more damaged components. In tissues under constant demand, liver, muscle, blood vessels, brain, biological friction begins to rise. It is like a city whose waste trucks still move, but less often. Streets remain open, lights still work, but accumulation has already begun. This is why autophagy matters. Not because it sounds exotic, but because the body is always producing material that must be cleared. Cellular health depends not only on building new parts, but on removing old ones. Health is partly construction, it is also sanitation. Source: the master switches. This chapter explains the biological root causes that regulate autophagy. Autophagy does not rise because a food is labeled healthy. It responds to internal signals. Two of the most important are AMPK and mTOR. AMPK acts like a cellular fuel gauge. When energy is low, it helps initiate autophagy. mTOR behaves differently. It senses abundance, amino acids, insulin, growth signals, and when active, it generally suppresses autophagy because the cell interprets the environment as one for growth, not recycling. This is why fasting is such a powerful inducer. Nutrient availability drops, insulin falls, AMPK activity rises, mTOR signaling is reduced. The cell shifts from storing and building toward repair and reclamation. Food complicates that picture. Eating usually signals abundance, not scarcity. So when people say a food activates autophagy, the claim is often too blunt. What science actually suggests is more nuanced. Some food-derived compounds may influence the pathways that regulate autophagy, including AMPK, mTOR, SIRT1, oxidative stress, and lysosomal function. That is not the same as saying a meal reproduces the physiology of fasting. Source. The difference matters, because in biology, a pathway can be nudged without the whole organism entering the same state, the cellular machinery. This chapter explains how the autophagy system itself works. Autophagy begins when the cell identifies material that is no longer useful or no longer safe. A membrane starts to form, like a soft-walled container rising around old cargo. This structure expands, encloses damaged proteins or worn mitochondria, and becomes an autophagosome. It then fuses with a lysosome, the cell's acidic recycling chamber. Their enzymes break the contents down into smaller units that can be reused. It is not destruction for its own sake. It is selective dismantling in service of survival. This system is deeply tied to nutrient signaling. In low-energy states, autophagy helps cells conserve resources and maintain quality control. In aging or metabolic overload, impaired autophagy is linked with the persistence of damaged cellular parts, increased oxidative stress, and reduced resilience of tissues over time. Still, the pattern is clear enough to ask a careful question. Which foods contain compounds most often discussed in the cellular conversation? 5 foods, 5 signals. This chapter explores 5 foods commonly linked to autophagy-related pathways. The first is green tea. Its best-known compound, EGCG, has been widely studied for its ability to influence autophagy-related machinery, including Beclin-1, LC3, P62, AMPK mTOR balance, and lysosomal activity. When in research settings, it appears less like a simple stimulant and more like a messenger that changes how cells respond to stress and cleanup. Source. The second is turmeric through its major polyphenol curcumin. Curcumin has been studied for effects on ACT/mTOR and related signaling pathways involved in autophagy. It is frequently described as a modulator of cellular stress responses, inflammation, and recycling pathways, though bioavailability in humans remains a major limitation. Source. The third is red grapes, often discussed because of resveratrol. This compound has long been examined as a calorie restriction mimetic, partly because it interacts with signaling networks tied to autophagy, including SIRT1, AMPK, and mTOR-related pathways. Source. The fourth is broccoli sprouts and other cruciferous vegetables, are rich in sulforaphane. Sulforaphane is studied not only for detoxification and oxidative stress responses, but also for its potential to influence autophagy-related mechanisms in experimental models. Source. These foods do not operate like 5 identical keys. They are 5 different chemical languages spoken to the same cellular machinery. Myth versus mechanism. The myth says, eat the right food and autophagy turns on. Science says something more restrained. Yes, dietary compounds can modulate autophagy-related pathways. Reviews consistently support that broad idea. But modulation is not the same as guaranteed activation across the whole human body. A compound may influence one signaling route in one tissue, under one condition, at one dose in one model. That is very different from claiming a plate of food reproduces the organism-wide response seen during fasting. Source. There is another complication: bioavailability. Many compounds that look powerful in laboratory studies do not dissolve well, are absorbed poorly, or are rapidly broken down in the human body. Curcumin and resveratrol are classic examples. Their molecular promise is real. Their journey through digestion, blood, liver metabolism, and tissue delivery is far less straightforward. Source and autophagy itself is not always universally beneficial in every tissue, every disease, or every amount. Biology prefers balance. Too little cleanup allows debris to accumulate. Too much or poorly timed activation can also create strain. The body is not asking for a permanent state of internal demolition. It is asking for regulation. So the honest scientific version of the title is this: some foods contain compounds that may influence autophagy-related pathways, but none of them act as a simple substitute for fasting. Reading the body's signals. This chapter explains what bodily signals may biologically represent. The body does not provide a direct sensation called autophagy level. There is no receptor in the skin for lysosomal flux. What the body does provide are broader metabolic signals. Reduced resilience to nutrient overload, impaired recovery, increased biological inflammation, and age-related decline in tissue function can all exist in landscapes where autophagy regulation is relevant. But these are shared territories, not unique signatures. They belong to many systems at once, and perhaps that is the real lesson. While cellular damage is rarely theatrical, it is sedimentary. It settles in layers. It builds in silence. It alters function gradually, then visibly. By the time a tissue begins to struggle, much of the story has already happened at the level of proteins, membranes, and organelles. Autophagy is one of the systems trying to keep that story from becoming larger. It is the body's quiet sanitation department, a recycling river moving through every cell. The biological answer is quieter than the headline. Certain foods, green tea, turmeric, red grapes, cruciferous vegetables, and spermidine-rich foods such as wheat germ, contain compounds studied for their ability to influence autophagy-related pathways. But food does not flip this system like a switch. It interacts with it, nudges it, and, in many cases, only partially and contextually. Must source see source. What remains true is this. The body is always cleaning, always sorting, always deciding what to keep and what to dismantle. Long before disease becomes visible, cells are already negotiating stress, waste, and repair. The body gives signals before breakdown. Health is not sudden, it is cumulative. Understanding the body is the first step to respecting it.